The word to be presented below is named the omics era, protemics, importance in cancer research. The members of this work are VALLEJO-CARDONA, Alba, Adriana, ROJAS-CERVANTES, Karen, Olivia, VERDUGO-MOLINARES, Maritza, Guadalupe, and LIMON-ROJAS, Areli, belonging to the Center of Research and Technological Assistance of the State of Jalisco, located in the city of Guadalajara, Jalisco. What is the cancer? The cancer is a pathology that leads the cause of death in the population worldwide. Also, it reports this increase when enhancing of life expectancy. When we talk about cancer, we refer to conditions characterized by abnormal cellular behaviors outside of biological controls. All cancer has three characteristics: uncontrolled cell division, invasion and destruction of normal tissues, and colonization of distant sites, being able to survive in external environment of its origins and continuing its invasion. Knowing the tumor microenvironment of cancer is very important for its study, bringing innumerable advantage. For example, knowing the origin of cancer is not the only option for the development of biomarkers and therapeutic targets. It's also important to study the tumor microenvironment as well as the mechanism involved in one of the most critical parts of cancer, the metastasis. By knowing the biological of the different stages of cancer, it will be possible to detect in the early stage and therapy avoid its metastasis. Being able to characterize biological systems will lead of personalized medicine, that is, treating cancer according to its type, its location, its state without affecting normal cells. Even the recent advances in treatment and diagnosis methods, most of them are invasive and inefficient in a lot of patients. It is still a change to expand and get better options for the patients. Therefore, it's fundamental to continue with the research for a new biomolecular or molecular change that can work in diagnosis, prognosis, or treatment of cancer. Being able to know this biology is possible through the study at the molecular level. For this is necessary to resort to the omic techniques for this study. The investigation of omic science is referred to the use of technologies to analyze biological systems at a molecular level. These techniques are capable to reproducing data that correlate with a specific state of biological systems. The molecules that the omics study give the name are metabolomics, proteomics, genomics, and etc. An important fact in the omics is having the capacity to recollect useful information about the biological system. This recompilation is called multi-omics. Multi-omics consists of the approach of the data generation of the omics, creating a big scale database, giving the possibility to make a big analysis of this information and use it to understand the biological system. This can be focused on looking at data about the specific disease in this physiopathology and proteins, genes, metabolites, and others, and their interaction relate to the specific events. We can mention a lot of sites of database that integrate omic science, such the cancer genome atlas, the human proteins, and Biof, gene bank, catalog somatic mutation, and all of the rest. Each one of them has provided below information to find auto mechanisms of metastasis, resistance to drugs, metabolic pathways. And in all of this omic science, it is important to mention one that have given different biomarkers and therapeutic targets that are widely used in clinical practice. And it is necessary to personalize medicine, the proteomics. The proteomics is the molecular science in charge of studying the proteome of any biological system. These include the proteome of the structure of proteins, their function, interaction and modification in any situation, using quantitative measurements to characterize any biological process and these separate mechanisms. The proteome of a living system provides fundamental details in understanding of this biology and how this biology is altering the face of certain specific physiological or pathological condition. The proteome of a living system provide fundamental details understanding out of this biology and how this biology is altered in the face of certain specific. The proteome tends to change in response to modification of this normal condition, providing fundamental information for the study and any pathology. Within the size, use of protemics allows early diagnostic, prognosis, and monitoring of the disease. It's also necessary for development of drugs. It's allow the identification of therapeutic targets or biomarkers. Proteins are thought to drive tumor growth, tumor invasion, and respond to target therapies. So identify change in protein signaling helps understand the mechanisms of cancer progression. Cancer proteomics basis is a study on the quantification and analysis of protein concerning the healthy counterparts for the identification of information that helps molecules in biological process. It can be used as characteristic molecule of different stages of cancer. This study of its structure, function, and quantification could improve patient outcomes. This characteristic is what makes them key molecules of this size can act as a biomarkers or therapeutic target for its treatment. The study of proteins, various biological systems are used in which are trained to extract the proteins so that they can be analyzed. We will describe the most important characteristic of cell lines, xenograft, and patient tissues. The cancer lines are useful of the protein study cancer in vivo, allowing a better understanding of cancer mechanisms in basic biology science. For the most part, they retain the same characteristic as the tumor of origin. The use of cell lines present easy manipulation and characterization and high reproducibility. In addition to the fact it is not necessary to obtain ethical permits for this use. And however, any inadequate quantitation during this culture of extraction proteins will result in a loss in a morphology and to impair the proteomic characterization. Cancer cell from cell line culture or cell from cancer patient can be grafted into most models generation xenografts. Xenografts represent a greater similarity to the mechanisms of cancer in humans. However, the environment in which the tumor develops varies significantly. In addition to this, it's necessary to have ethical permits for the manipulation. The proteomic analysis of patient can be analyzed with a sample of tissues, and or the tumor has developed, but is in invasive analysis for the patient. In some occasions, it is difficult to obtain so biofluids such as blood, tears, cerebral pain fluid, prostatic secretion, and others have also made it possible to identify clinical relevant proteins of the disease. Versatility in the use of sample is a crucial point. Currently, the characteristic and advantage of using these fluids replace tissues. Biopsies have been analyzed and liquid biopsies have been determined. This option allows cover all aspects of cancer. Unlike tissues biopsy, liquid biopsies could be used for early detection and diagnosis, prognosis, and monitoring. The procedure consists of isolation in ties derived from the tumor from these samples, such as circulating tumor cells, circulating tumor DNA, or extracellular tumor vesicles. Finally, two conclusions. The proteomics is an omic science that has revolutionized cancer studies by extending and existing knowledge, eluding raw mechanisms in basic biology. And based on this, applying it on the critical care of patients. Personally, medicine has been largely on the study of proteome science. It allows the identification of biomarkers and therapeutic targets to direct a treatment according to the type of pathology and level which the patient is. And in this way, minimizing the side effects of current chemotherapies. Proteomics allow the studies of variety of samples and models, and has provided a greater understanding of the mechanisms which say pathology is addressed. In addition to being able to identify the main function that certain cells have in the behavior of metastasis or resistant to treatment drugs, which has allowed important advance in adequate treatment of patients. Although in some cases the disease is not eradicated, the survival time has represented a significant increase. And the last one, proteomics is important for the collection of optimal data. Thank you so much.